Hello, everyone. This is a review of exam 3, which is probably going to be pretty long since there's a lot of material to cover. So let's just get started. So a first key topic to introduce you to all of the material on this exam is that the gene proper region tends to be conserved because specific proteins are critical for different functions, but the cis regulatory switches can be variable in their instruction making. So they can vary in the amount of protein produced, the location in which it's produced, and the time during which it is produced. Which brings us to our first main topic about duplication of loci. So this allows for expanded range of functions. And a few terms. Homology is the similarity in structure across different species due to shared common ancestry. So many animals have descended from a common ancestor, and you can compare some of their bones in their limbs, for example, and you can match up the same bones. They'll often have very similar shapes, but of course the limb shape itself is different depending on that species form and its body plan. And an example of duplication that we covered is with the early the globin gene. There was a duplication that occurred so that there was an alpha chain gene and a beta chain gene. And so we can compare these genes and look at the orthologs and the paralogs. So orthologs are DNA sequences with very similar sequences. The protein that is produced is very similar and it follows speciation events. So these genes are related to different another gene in a different species due to descent from common ancestors. So you're looking at a gene that's very similar to a gene in a different species. And that helps you understand their descent from a common ancestor. Now, paralogs are genes related to another gene in the same species. So that can also help you understand the genetic variation of genes within the same species. And then if we're comparing all of these organisms and the loci, we're looking at homologs, which are DNA sequences that are alike. So more of a loose likeness than orthologs or paralogs. So remember, paralogs is the same species, orthologs are different species, and homologs are more of a general term to describe sequences that are alike. And that goes back to our definition of homology. Adding on to duplication, an issue that arises when a gene is duplicated, caused by a duplication mutation, is you have something called functional redundancy. So you have now two loci that are doing the exact same thing. And whole genome duplications, which are very common in plants, can actually be beneficial. And we see this a lot in a lot of crops that we consume. But in other cases, since duplication is a gain of function mutation, it's actually harmful or toxic. So we know that many genes are genes that have arisen as duplicates of a different gene. But in that case, many of them are not harmful. So how did that functional redundancy resolve? And that brings us to two different models, Ohno's model and the DDC model. So let's start with the DDC model on the right, just because it's fully in frame. So DDC stands for duplication degeneration in a complementary way. And there are definitely some key concrete differences between these two models that hopefully can help you get them straight, because there's a lot of information here, but they're pretty different. So in the DDC model, before the duplication mutation occurred, the genomic locus that we're dealing with has had a mutation occur that develops a new function. So it has a gain of function mutation. Now it has more than one function. Then after duplication, the paralogs that are created by this duplication, they essentially divide the functions between themselves. So we need a gain of function mutation to occur before duplication in this model so that we have different functions that can be divided between the different paralogs. But it's important that both paralogs are retained. And an example of this that we talked about was one of the monkeys that I believe had like a separation of digestion of different pHs of like leaves or something. So in that case, they had paralogs, and one of them handled food at like a lower end of a pH range, and the other one handled leaves with a pH of the upper end of the range. So that's duplication degeneration in a complementary way. So if you can remember that DDC stands for that, you can remember that the paralogs have to complement each other in terms of function. And then Ohno's model. So in Ohno's model, duplication occurs. We don't have a gain of function mutation before that duplication. And remember, duplication is also gain of function. But here, duplication just occurs, causing immediate functional redundancy. So now we're currently at a state where we have paralogs both doing the same thing. And again, it's critical that one paralog continues to carry out the original function. Otherwise, we would have some sort of loss of function associated with that, because we need to still produce the same protein. So functional redundancy here can be resolved in three ways. We can have a retention of ancestral function, we can have a gain of function mutation, or we can have a loss of function mutation. And those are pretty distinct. So the loss of function mutation occurring is called pseudogenization. This is where the extra paralog has a loss of function mutation, making it non-functional. Neofunctionalization, remember the prefix neo means new, so we would have a gain of function mutation at that extra paralog, giving it a new function. And then retention of ancestral function is when the extra paralog retains ancestral function. So we still have that functional redundancy, meaning this is gain of function. So you can see Ohno's model is a bit more complicated, but the key difference here is that in Ohno's model, there is no gain of function mutation before duplication. We just have a duplication mutation, and then three different scenarios can happen. We can either have, we can still have functional redundancy called retention of ancestral function, which is gain of function. The paralog created can have a loss of function mutation called pseudogenization, which makes it non-functional. So we have that original paralog continuing to carry out original function, almost as if the duplication didn't occur. Or we can have neofunctionalization, where we have the original paralog continuing to carry out original function, and then we have the extra paralog with a new function. But in the DDC model, we only have one outcome, which is that the paralogs split their duties or their functions between themselves. We talked about Hox loci, which are pleiotropic master regulators that have a lot to do with vertebra development along the AP axis, along the body plan. So these genomic loci encode transcription factor proteins that specify body plan along the AP axis, limbs, and urogenital system. And we learned two different terms here. Spatial collinearity is where the sequence of loci coincides with the order of development along the AP axis. So if a genomic locus comes before many other loci, then that will correspond with it affecting something more early on in development than the other loci that come after it. And then temporal collinearity is similar. This says that the sequence of loci coincides with the time of development. So a loci early on in a sequence in the DNA, that carries out its function earlier on in that organism's development, and the further along loci happen to be active later on in development. Next, we talked about transcription, gene regulation, and translation. Many of these things might be a review, but there also is a lot that is new. But just a review of RNA. So it's single-stranded. Instead of thymine, we have uracil, we have U. RNA has ribose instead of deoxyribose, and it's phosphodiester bonds for the nucleotides. And then we should be familiar with the different kinds of RNA. So we have mRNA, which is messenger RNA, transfer RNA, tRNA, and ribosomal RNA, which is rRNA. And you need to know the different RNA polymerases that correspond to this. So mRNA corresponds with RNA polymerase number 2. I like to think of this 2, the number 2 spelled TWO, and then another version of 2 is TO, and messengers deliver things to and from. So that's how I remember that. tRNA, I just remember T kind of rhymes, I guess, with 3. I don't know if it really rhymes. And then rRNA is just the one that's left out. Something interesting to note is that there are 61 unique transfer RNAs that carry those anticodons for translation. But there is degeneracy, as we might talk about it a little bit later. So there's 3 parts to transcription. That's when mRNA is produced. Initiation is where you have a promoter, which is a recognition site for those transcription factors to attach onto. So what happens is the transcription factor proteins are produced by a genomic locus. A good example is the Hox genomic locus. And it goes to the promoter, and the transcription factor protein binds to the promoter. This makes it a lot easier and allows for the RNA polymerase to bind to that sequence. So then it can transcribe mRNA. And when this happens, DNA is denatured into the open complex. So we have an open reading frame. Then RNA polymerase slides down the template strand, creating mRNA during elongation. So it is just matching up the nucleotides, the base pairs, making sure to substitute uracil for thymine when it matches the nucleotides to the template strand. And I might have a little diagram somewhere later on about this. And then termination, so a terminator sequence is reached, which causes the RNA polymerase and the mRNA transcript to dissociate from the DNA. Let's see what I got over here. This is just from Dr. Chu's study guide. Just extra details. Feel free to pause this video and read those. I'm just covering more of a general sense of what's on the exam. Another key topic that we talked about are gene regulatory networks, or GRNs. And again, the Hox genomic loci are great for this. So Hox genomic loci produce transcription factor proteins. And then these transcription factors bind to promoters, which can be enhancers or repressors, turning on transcription or turning it off. And you can see here in the top example in blue, so we have a transcription factor protein called HOXA13TF, transcription factor, binding to the regulatory switch, which in this case is an enhancer. This locus is HOXD13. Because the regulatory switch is an enhancer, it turns HOXD13 on so that the transcription factor of HOXD13 can be produced. And then things can get kind of complicated from here, moving down to the area in red. This is Sonic Hedgehog, which is a very silly name. I don't know if I would choose to name a locus Sonic Hedgehog. Like, I don't know. I can't even think of what I would name a locus if I were to discover it. But I don't know that Sonic Hedgehog would be the first thing that came to mind. I'm sure that the scientists had a good reason for it. I don't know if you could have a good reason for it. Anyway, moving on. Okay, Sonic Hedgehog. So again, HOXA13 transcription factor in green binds to Sonic Hedgehog enhancer, turning Sonic Hedgehog on. But we also need the HOXD13 transcription factor to bind to the promoter. And then from this, so we've got two enhancers here and two transcription factors binding to those enhancers. Then that turns Sonic Hedgehog on so that it can produce the Sonic Hedgehog signaling factor protein. And in the purple area, so in this case, we have a silencer regulatory switch, and we have a repressor protein. In this case, it is actually HOXA13 acting now as a repressor protein binding to the silencer, turning HOXA11 off. But because we have Sonic Hedgehog as a signaling factor, which binds to an enhancer regulatory switch on the purple HOXA11 site, then HOXA11 is able to produce its transcription factor protein. So you can see that for loci, there can be many pieces and parts. to it. It's not always just one enhancer or one silencer. There could be many different factors affecting whether or not transcription is turned on or turned off for that specific locus. And you can also understand that mutations here can really make a lot of things fall apart. Because if you were to have a mutation of HOX A13 causing it to be non-functional, then this whole thing falls apart because that transcription factor cannot bind to turn on transcription for other loci, and then those loci and their corresponding proteins can't go out and also bind as well. And there's many different cases of transcription factor proteins interacting with cis regulatory switches. In the top case, we have an enhancer cis regulatory switch before a promoter. So remember, cis regulatory switches are the instruction givers. So when we have an activator protein binding to the enhancer, it turns on mRNA transcription for this locus. Conversely, a silencer cis-regulatory switch turns transcription off when a repressor protein is bound to it. So remember, activator proteins bind to enhancer cis-regulatory switches, and repressor proteins bind to silencer cis-regulatory switches. And enhancers cause mRNA transcription to be on, and silencers, they tell it to be off. Now, sometimes an activator protein needs a co-activator in order to bind to the enhancer. And also likewise, sometimes repressor proteins need a co-repressor to bind to the silencer. So they need a little something extra to be able to bind. You can have mediator proteins that act as bridges between the enhancer cis-regulatory switch or the silencer cis-regulatory switch and its corresponding bound transcription factor. And these are to help it get closer to the RNA polymerase, to speed things up, to essentially just get the instruction maker closer to the action. Another thing that might be required for a transcription factor to be able to function is a hormone. So a hormone might need to bind to a transcription factor so that transcription could be turned on or off, depending on what transcription factor and cis-regulatory switch you have. Other times, you need more than one transcription factor, and these are called homodimers. So two transcription factors coming together, either acting as activators or repressors. And other times you need some sort of chemical modification to the homodimer or transcription factor before it can be active. And a common one is phosphorylation. So you can see there's a lot of different ways for transcription to be controlled. And likewise, there is a lot of variation that can happen all throughout these different processes. So here's just some examples of areas that can have single nucleotide polymorphisms, SNPs, just single base pair changes. In the non-coding region, you could have a SNP at the 5' untranslated region or the 3' untranslated region. It could happen in the introns, intergenic regions between these genes. In the pseudogenes, it can happen in splicing areas, and in the transcriptional and translational regulators. And encoding sequences, so we can have a synonymous or silent mutation, or we can have a functional mutation, which this is where there is an amino acid replacement, but due to the nature of a specific protein, that amino acid replacement allows for the same function. We can also have a non-functional SNP, of course, that causes the protein to behave in a different way, be non-functional perhaps. So that would be a deleterious mutation. It could be. We talked about RNA processing. So in eukaryotes, there are a few modifications that have to be made to the mRNA before it can leave the nucleus and go to the ribosome to be translated. And prokaryotes, the whole central dogma is simpler, for lack of a better word, because, it definitely is complicated. But so for mRNA, you need to add a 5' cap, a 3' poly A tail, composed of approximately 100 adenines. Splicing must be performed where you cut out the introns and you join the exons. And sometimes reverse transcription can occur, making processed pseudogenes. So I have pictures to go with these. So for DNA, remember DNA is double-stranded. The 5' to 3' end is the sense strand, and the 3' to 5' end is the antisense strand or the template strand, where the mRNA has corresponding base pairs to. And the DNA has exons and introns. Remember, if we are looking at mRNA, introns are flanked by GU and AG. So that's how the spliceosome knows how to cut those out. So when we're processing mRNA, first we have to add that 5' cap. We got to remove the 3' end and add a poly A tail. And then we have to splice it all together. So we cut out the introns, going by the GU and AG as our markers to know where to cut. And then we join the exons all together. And then from this, our mRNA can be used for translation. Yeah, again, each intron begins with a 5' GU and it ends with a 3' AG. This consensus sequence, which is shared across every organism, so it's highly, highly, highly conserved, is recognized by the spliceosome, which is what splices the mRNA. And I have another little diagram here where there's a lariat loop formed when we're splicing out the intron. And again, it's critical that this area be conserved, be accurate. Otherwise, there could be a frame shift mutation if a part of the intron is left in the sequence, or if a part of an exon is cut, for example. Then, of course, the sequence of nucleotides and corresponding codons is completely messed up. So you'd have a frame shift mutation resulting in a non-functional protein. Reverse transcription is when we have an mRNA, normally it goes to the ribosome for translation to produce a protein. But in this case, we have this little guy called reverse transcriptase. And it basically just takes the mRNA and replicates it and inserts it back into the DNA sequence. kind of like a cleaner crew, but if you like, if they clean up your art projects and you lost all your work, like kind of bad news. And this can be bad because this would be gain of function. Since mRNA has been processed, we've taken out the introns. So there's no introns, meaning there's faster transcription, which this can actually lead to cancer because we're creating, we can create proteins faster and faster. But luckily, most times, these processed pseudogenes have loss of function mutations, making them non-functional, and that's a good thing to prevent the chaos and the mess. This is the genetic code table. We've all seen it. Pretty easy to read. Just look for the first base on the left, go up to the top, look for the second base, and then the third base on the right. Here there's 61 codons, but only 20 amino acids, meaning we've got redundancy in the genetic code. So for translation, which happens at the ribosome, in the cytoplasm outside of the nucleus, we have our little tRNAs, or transfer RNAs, acting like little storks bringing the anticodon babies to match up with the mRNAs. And they're carrying along corresponding polypeptides, or not polypeptides, sorry, amino acids. And these amino acids link together into polypeptides. And remember, so mRNA starts with the AUG that starts the sequence of amino acid chain formation. So the tRNA will keep adding anticodons and delivering amino acids until it reaches a stop codon, and then it pops off and you're left with the polypeptide, your protein. And then these proteins can be modified to be allowed to go wherever they need to go to perform their function. Now, because the genetic code is degenerate, we have many different codons, but only 20 amino acids. It's the case where if you look back to the genetic code, often you'll see this degeneracy in the third position of the codon. If we look at threonine, for example, you'll see that all of them start off with AC, but then we could have at the third position a U, C, A, or G. So it could be any one of those. So we have the wobble rule, where even though the first two positions strictly follow the base pairing rule, the third position of mRNA can wobble or tolerate mismatches. And this actually corresponds to the first position of transfer RNA. And so silent substitutions can occur here. So going back to threonine, if we had it to where originally our sequence would have been an ACC, but we had a synonymous mutation changing it to an ACA, yeah, a mutation would have occurred, but there's no change in amino acid due to the degeneracy, because we're still producing the same amino acid. So that's very helpful. Next is alternative splicing. The benefit to alternative splicing is that one single genomic locus has enough information to make several distinct protein products. This can be cell and organism specific. But something key to note is that you always have to have the full-length protein product. So you always have to produce that original one or else you'll have loss of function. And this picture's not very good, so I'm going to move on to the second one. So here we have different exons and different introns, and we're going to splice them, but because we know that the spliceosome looks for GU and AG, there is some variability in where it can actually splice. So here in the on the left case, we had exon 2 being skipped. So what it did was for the first intron, it saw a GU here, but it actually skipped the first intron's AG, and it actually went to the AG of the second intron. So it treated exon 2 as if it were just a continuation of the first intron. So exon 2 on the left was spliced out. So this led to a specific protein being produced, different from what would have happened had we had just regular splicing. And on the right, we have exon 3 being skipped. It too was treated as if it were part of an intron due to how the spliceosome reads GU and AG. And this produces another different protein. But again, you must always have the full length protein product. So you have to have a protein product where you're not splicing out any of the exons, where you would have exon 1, exon 2, exon 3, and exon 4. But again, the good part of this is that we can produce multiple different proteins from the same locus. So that gives organisms a lot more freedom, a lot more different things that they can do. So alternative splicing can happen when a splicing repressor prevents recognition of the 3' splice junction. So the splicing repressor binds to that AG area, the first AG that the spliceosome might come across, and so it can't actually splice at that area, so it has to go to the next AG. And you can imagine we could have several different repressors which would cause the spliceosome to not be able to get to an AG for a long time, causing many different exons to be cut out. But on the other hand, sometimes splicing enhancers are needed to make the full-length mRNA. So sometimes alternative splicing is actually easier to do, and we still need the full-length protein product. So we need splicing enhancers, which promote recognition of those GUs and AGs so that the spliceosome can stop splicing things out that it wasn't supposed to have spliced out. This means that introns can act as exons and vice versa. So we have a lot more flexibility and freedom in the genetic code, or not the genetic code, our genetic sequence. And, this can happen in a variety of different ways, because again, we only have for mRNA A, C, G, and U. So there could be many different GUs. The question is which is the right one to start off with if the spliceosome is splicing an intron out. So you can see there can be a lot of flexibility in terms of what the spliceosome can do. But it really is critical that we still produce the full-length protein product. So why is this important? Why is alternative splicing important? So alternatively spliced sequences, the protein products made from them can vary in function, and it can cause cell type differentiation. In this example, one sequence affects smooth muscle cells and the other one affects striated muscle cells. So we have a kind of muscle cell differentiation. In this case, for the same genomic locus, we're just producing alternative spliced versions when we're cutting the introns out, which produce different protein products. And then constitutive exons, these are exons that have critical information that really need to be kept in the sequence. So these are exons are present in all variants, all of the alternatively spliced variants. Alternative exons are more flexible, so they're not always included. Their function is not significant enough to warrant them always being in a sequence. And then our last big topic was about Hardy-Weinberg and genetic drift. But remember, we said that there were only going to be 3 or 4 questions on the exam. So it's probably critical that you just at least know how to problem solve these questions and you know the formulas. And there are a lot of helpful YouTube videos on this if you need a refresher. So a few different equations. You need to know what allele frequency is. So this is just the number of copies of a specific allele in a population over the total number of all alleles of a gene in a population. Genotype frequency is the number of individuals with a specific genotype in a population over the total number of individuals in that population. The Hardy-Weinberg equation can be used to find genotype frequency. It's p squared plus 2pq plus q squared equals 1, where p squared is the genotype frequency of homozygous dominant individuals, 2pq is the genotype frequency of heterozygous individuals, and q squared is the genotype frequency of homozygous recessive individuals. And maybe you might be able to make some sense of this beyond just memorizing the equation. So when we say homozygous, we're talking about the same allele, right? So p squared just means p times p. So we have the same allele. Same thing with q squared. And then, so for heterozygous, we have 2 different types of alleles. So we have p and q. And then p plus q equals 1 is the allele frequency, where p is associated with the dominant allele frequency and q is associated with the recessive allele frequency. So you need to use both of these equations often alongside each other to solve for p or q when you know one or the other. And Hardy-Weinberg equilibrium occurs when 5 assumptions are met. First being that you have a large population. Second is that there's no migration, nobody comes in, nobody leaves. Third is that there's no new mutations. Fourth is that we have random mating. And 5 is that there's no natural selection. We talked about allele fixation and elimination. Fixation means that 100% of individuals in a population have the same allele. And the probability of fixation in a population is 1/2n, where n is the population size. A good example of this is if we're talking about all of the human population in the entire world having the same allele, that probability is really, really, really low because n is so huge. So 1/2n would be such a small number. So we're like like it's pretty much impossible for all of us to have the same allele for a given gene. Another definition is genetic drift. So when a new allele arises, it's either fixated or lost in a population, which is influenced purely by chance. It's all about probability. So in contrast to a human population example, you could imagine that in endangered species or critically endangered species, individuals with like who are in a very small population, genetic drift is very powerful, and the new alleles can be fixed pretty easily, which can be detrimental because there's less flexibility for the phenotypes. Since the population is so small, and you might have closely related individuals breeding with each other, their variation, their genetic variation is reduced. Another equation alongside the probability of fixation is the probability of elimination, which is just 1 minus the probability of fixation. So 1 minus 1 over 2n. And then if we want to know how long it takes for a new allele to be fixed, so how many generations, that's just t equals 4n, where n is the population size. So in a bigger population, it takes a longer time. And we use generation over year because generations apply to every organism, because different organisms, they have different times in between their generations. The time it takes for many different animals to sexually mature can vary widely. So it's better to use generation. So for this, t equals 4n. Another powerful effect is the founder effect, where a small number of individuals from a population move to a new area, and this new population is isolated from the large population. So they're taking their alleles with them to start this new population. And the genetic variation of this founding group is, it is reduced because you are taking only a small group of individuals. So I think this should say smaller. So genetic drift here can be powerful, and fixation can occur as well. And the example we talked about was this colony of 15 Britons in 1814. One of the individuals was a carrier for an autosomal recessive disease causing blindness. Even today, among 240 descendants, 4 are blind and 9 are carriers. So this was caused by the founder effect and genetic drift. So we still see this, these alleles here within this population. And then I just have the questions that we went over in class. So if you want to look at these answers, feel free to pause the video we went over in class, and it's also on Blackboard. Yeah, so feel free to pause if you need any of this information. And then here is the summary of all the information. So again, pause this. And of course, you can rewatch this video, go back to the book, watch some of the recorded videos on Blackboard. to get more practice and more exposure to the topics that could be covered on the exam. So good luck.